Good evening, everyone, and welcome to week 10 of Basics of Crop Breeding and Plant Biotechnology live interaction session. I am Prashasti Pandey, PhD and PMRA Fellow from Department of Botany, Banaras Hindu University. And if you have attended previous sessions of these live interaction sessions, in these sessions, we will basically go through some questions of previous year's assignments and we will try to solve them, understand the concept of those questions. And hopefully, they will help you in solving this year's assignment of the course. Okay. So, with that introduction, let us begin with our session. So the first question for today is which of the following is correct for bidirectional promoters? The options are A, expression of a single gene in two different directions. B, a single RNA polymerase is used to transcribe both the genes, always less efficient than unidirectional promoters, and transcription of two different genes from two different RNA polymerases. I'll give you 2 minutes of time. If you know the answer, you can speak it out aloud or also you can write it in the chat box. So let's solve this question. Okay. So first, let's understand that what are bidirectional promoters. Okay. So basically, when we are talking about some gene, okay. So suppose this is a gene. Okay. That gene has to be derived by some promoter. Okay. Without the presence of promoter, the gene cannot be transcribed into its mRNA, okay, or it cannot form the functional part of the DNA to get processed further. So it always has to be derived by a promoter. Now, as the term suggests, that it's a bidirectional promoter. Okay, so by bidirectional, it means that the promoter can drive genes in two different directions. Okay, so that is the exact definition of bidirectional promoters. That promoter is located between two genes and it will drive the transcription of both the genes. Okay, so if this is your promoter, then the gene can be present in this direction also, and the gene will be present in this direction also. Okay, so the transcription will happen in both the directions and it is driven by one single promoter. It can be either simultaneously or it can be independently in opposite directions. Okay, so these kind of promoters are called as bidirectional promoters. Okay, either they can so sometimes these promoters can drive one gene completely and one gene partially, or they will drive both the genes in same at the same time, or they can be independently and 100% driving that particular gene. So uh, this is the definition of bidirectional promoters. Now how it works: so basically, in this case, bidirectional promoter will always lie between two genes, and each gene will be oriented in opposite directions because from the promoter, the transcriptional starting site should always begin. So, if for this gene the transcriptional starting site is here, for this gene it should be here. So that is why the gene direction for this gene will be in right side, and for this gene will be in left side. Okay, so the gene orientation in this case, in case of bidirectional promoters, will always be in opposite direction. Third thing is that this promoter now, so the function of promoter is to recruit the RNA polymerase so that the following transcription can take place. So this promoter will recruit RNA polymerase in both directions. Okay, it will recruit the RNA polymerase here as well as here also, so that the transcription can happen in both the places and in both the directions. So that is another property, that transcription will be in both directions. Now, the key features of bidirectional promoters are that it can control two adjacent genes, and the genes in this case should be very closely present. Okay, so they cannot be very far separated, or they cannot be some other site present between the promoter and the gene. Otherwise, the driving force of the promoter will not function properly. So the genes should be closely spaced. There should not be any other promoter or any other gene present between the bidirectional promoter and the gene that it has to transcribe. Third thing is that it can be co-regulated. Co-regulated means that both the genes can be driven or it can be transcribed or expressed together. And this is mainly found in eukaryotes and viruses. Okay. So this is a simple figure of a bidirectional promoter, that the promoter and both the sides, the genes will be there, gene A and gene B. So it can happen in both directions. So for example, this part is the promoter region. This is your gene 1 and this is the gene 2. Okay. So now your promoter can drive this gene in this direction and this gene in this direction because the transcription start site is here. Okay. And And now, for this particular promoter, the gene 1 and gene 2 is here. So the start site for this gene 2 can be present in this direction, and for the site of gene 1 can be in this direction. So in this case, the start site region will be overlapping, but in this case, it is not overlapping. Okay, so both situations can be present for driving the gene, and this is basically the function of a bidirectional promoter. Now, coming back to our question, that now we understood by now that bidirectional promoters will allow the transcription in both the directions and from the same promoter region. Okay, and typically this will mean that the two genes are placed head to head in opposite directions, and then promoter can drive the transcription in both directions. Now, let's evaluate our options. So, the first option was that expression of a single gene in two directions. So, yes, this was the exact definition of bidirectional promoters, that single gene will be derived. The expression of single gene in two different directions is also possible. For example, if the promoter is present here, the gene can be present on both the direction, and it can be same gene, but it can be derived from here and it can be derived from here. Okay, if the reading codon for both these genes are same, then the outcoming RNA from this will also be same. But in certain cases, even though the sequences are same, the reading strand, that is the 5 dash to 3 dash, the 3 dash to 5 dash reading strand in this case will be opposite to each other. So in that case, the mRNA that will be produced will not be the same, even though the gene is same, but the resulting mRNA will be different. So this option is correct. Option B was a single RNA polymerase is used to transcribe both genes. No. So even though both the genes are being regulated by a single promoter, the there needs to be two different RNA polymerases recruited for the transcription of two different genes. It is not possible for one single RNA polymerase to perform transcription in both the directions at the same time. So B statement is wrong. C statement is always less efficient than unidirectional promoters. Not true, because there are sometimes efficiently will depend on various contexts. Okay, depends on how strong the promoter is, how efficiently it is able to regulate that particular gene, on what all conditions is it getting activated, and there are multiple factors which can determine the efficiency of the promoter. So C statement is wrong. D is transcription of two different genes from two different RNA polymerases. So, so in this case, option D will be our correct option. That transcription of two different genes from two different RNA polymerases. So even though option A is also slightly correct, but the entire statement does not describe what meaning should be correct. So it will be wrong in this case. B is also wrong. C is also wrong. So option D is the correct answer. The transcription of two different genes from two different polymerases, that we just understood from our figure, that if this is our promoter and we have one gene in this direction and the other gene in this direction, then the D option is saying the transcription of two different genes. That is, if this is our gene 1 and this is our gene 2, then there will be one RNA polymerase recruited here and one RNA polymerase recruited here. So this transcription will be in this direction and this transcription will be in this direction. So transcription of two different genes from two different polymerases is correct for bidirectional promoters. Okay, I hope this question was clear. Let's move on to our next question. The size of the gene X primary transcript and matured mRNA is same. What might be the possible justification of the statement? Uh, Option A is during splicing, the introns are removed and the addition of poly A in the 3' end makes up the size as primary transcript. Option B, the gene contains no introns, hence the size of primary transcript and matured mRNA is same. C, splicing does not occur in this gene, hence introns remain in both primary transcript and matured mRNA. And D cannot be justified. Okay, so let's understand this question. So the question is talking about two different things. Okay, one is gene X, which is the primary transcript, and the other one is mature mRNA. Okay, and it is saying that the size of these two things are same. Okay, let's understand this thing with figure first. When we talk about a gene, okay, so DNA or the gene will have two things. It will have both exons as well as the introns. Okay, and introns are the intervening sequences. Okay, so intervening sequences, that means they are not required in the matured mRNA. So before the gene gets converted, before the pre mRNA or the immature mRNA gets converted into a functional mRNA, these introns are basically cleaved out and the exons are joined together by the process called as splicing. Okay, splicing, because our DNA sequence will have the codes of the introns as well. So during transcription, these introns are also basically transcribed in the mRNA, but this mRNA is immature mRNA. So once the process of splicing takes place, all these introns get cut off from their sites, and all the exons are then later joined together to form a matured mRNA. And this matured mRNA now will get translated to form our protein of interest. Okay, so this is the basic concept of gene and mRNA. Now this in our question, so the primary transcript word that was used is basically our pre-mRNA. Okay, so pre-mRNA will contain exons as well as the introns, in which exons are the coding regions and introns are the non-coding regions. And during the processing, which is called as splicing, so introns are removed by this process called as RNA splicing. And there is one more process that happens along with the removal of introns, that a poly A tail is added at the 3' end. So if this is our mRNA with removed introns, then in that case, towards the end, there is a poly A tail which is added. Okay. So there is a poly A tail added towards the end of the 3' region, and now this will be labeled as a mature mRNA. So that is why the pre-mRNA will always be longer than the mature rRNA. So because that is because the introns are getting removed, even though poly A tail is added, but the sequence size of introns is way huge than the poly A tail. So despite the addition of few A nucleotides towards the end, 3' end of the mature RNA, the mature RNA size will always and always be shorter than the primary transcript RNA. Okay. So now coming back to our question, option B is in this case is correct because, okay, let's talk about the options only. Option A was that during splicing the introns are removed and the addition of poly A tail in 3' and makes up to the size. So now why this option is wrong? Okay, even though in while forming the mature RNA, as I previously mentioned, that even though multiple A nucleotides are added towards the 3' end of mature RNA, this size will not be equal to any size of the introns. Okay, poly tail is usually 16 to 18 nucleotides, but introns can range from 50 base pair to sometimes 300 base pair as well, and they can even be longer. And not just single mRNA, there can be sometimes multiple intron regions. Okay, so that is why addition of poly A tail cannot be compared with the removal of intron. That is why option A is incorrect. Option B was that the gene contains no intron, hence the size of primary transcript and matured mRNA is same. This can be true. There are situations, there are certain genes in which the, the DNA sequences do not contain any intron sequences. All the sequences are already exon sequences. So now, when the DNA is converted into pre RNA, this pre-RNA will have all the exons already joined. So even after splicing process, because there is no all the exons are already joined. So even though there are no intervening sequences, only couple of A nucleotides are added towards the 3' end. That is why there are chances that the size of pre-RNA and mRNA will be same, because there were no intervening sequences to begin with in the DNA. So that is, in this case, the size of our gene X, which is primary transcript, and matured mRNA will be same. Now, option C was splicing does not occur in this gene, hence intron remains both primary transcript and matured RNA. This is not possible. The reason is that if the splicing will not take place, the pre-RNA or the pre-primary transcript will not get converted into mature RNA. So it will not become functional. So that is why this option is also incorrect. So this cannot be the reason why primary transcript and mature mRNA is same, and these cannot be justified. Of course, that is wrong because it can be justified. The justification is that there were no intron sequences in the primary transcript, and therefore, when it gets converted into mature RNA, there was nothing to cut out, and that is why the size of the primary transcript and the mature mRNA is same. Okay, I hope this was clear. So our next question is, which of the following is essential for successful cloning of a PCR product in TA plasmid vector? So the options are a vector with blunt end, a vector with 5' end containing T overhang, a vector with sticky end made by restriction digestion, and a vector with 3' end containing A overhang. Okay, so let's understand this question. It is very easy. This is a type of cloning method, basically TA cloning, which requires a TA plasmid vector. Okay, so let's understand what this TA cloning first relies on. Because there is no use of restriction enzymes in TA cloning. So, TA cloning will basically rely on the property of certain DNA polymerases, like the most common polymerase that are used in cloning is the tag DNA polymerase. Okay. The tag DNA polymerase has a property that after synthesizing PCR, every product in the PCR cycle, it will add a single A nucleotide at the 3' end of the PCR product. Okay, so so if this is our PCR product, that the original PCR product, and this is our 5' to 3' side and 3' to 5' side. Okay, so what this will do is it will add one A in this direction and one A in this direction after the cycle in the PCR. Okay, so the the resulting PCR product will have two overhangs of A on each side. So now, because there are single nucleotide overhangs, they can be easily attached to the complementary sequences. Okay. So now our PCR product looks something like this. This is the PCR product, and it has an A site here and an A site here. So what will be complementary to A? It will be T, right? So if it gets the T site in those both these directions, then in that case it can be easily attached. Okay. So now where does it get this T site from? So this T site is attained from the vector. Okay. And these 5 dash T overhangs are basically present on the vector, which will be used for the TA cloning. So the concept is basically allowing the base pairing of A and T, and this ligation is effective without the use of any kind of restriction enzyme. Okay. So the basic concept of TA cloning, if you look at this figure, is that this is our PCR product, okay? And our PCR product will have T overhangs in both the 3' direction, and our vector on the same place will have, the PCR product will have A overhangs and the vector will have T overhangs, okay? And when we ligate this A and T will bind together, and this entire entire plasmid will get converted into the circular plasmid, and it will be ready to get transformed. Okay, this is the figure, which hopefully will make it easier to understand about the TA vector. Okay, so this TA vector, this is the 3 dash. So this is the T overhang, and this is the A overhang. Okay, and by using T4 DNA ligase and the reaction buffer, this A and T will basically form pairs, and that is how insert is ligated into the TA vector. Okay. Now, coming back to our question, talking about the options that were given. So, for a successful cloning of PCR product, a vector with blunt end, no. If a vector is blunt end, there will be no base pairing between T and A, and the concept is the presence of overhangs of T and A in insert and vector. So, option A is incorrect. Option B was a vector with 5 dash end containing T overhang. This B option is correct because the vector will contain T overhang and the insert will contain A overhang because of the addition of an additional A during the PCR synthesis by utilizing polymerase like Taq polymerase. Okay. So option B is correct in this case. C was a vector with sticky and made by restriction digestion. So in this case, there is no requirement of a restriction enzyme which will digest and form the sticky end. So it is wrong. And option D was a vector with 3' end containing A overhang. This is wrong because vector will not contain A overhang. Instead, the PCR product will contain A overhang and the vector will contain T overhang. So option B in this case is correct. Okay. So the next question is, in a construct A, GFP was under promoter P. Another GFP construct generated using deleted promoter region of 500 base pair from P resulted same expression of GFP in a construct A. What can be inferred from this experiment? Options are: the region is responsible for coding gene, the region is essential for expression of genes, the region is irrelevant for the gene expression, and the region promotes RNA splicing. This is a little bit of tricky question, but you can try attempting it. So let's solve this question. Um, just there need you need to have a small understanding to solve this kind of questions. Okay. So in this question, this question is basically an example of an experiment called promoter deletion assay. Okay. So in this case, a part of promotion is basically deleted, and then it is studied that what effect will it have on the gene that it is transcribing. So the question says that in a construct A, GFP was under promoter P. So if let's say this is promoter P, and then this gene under its control will be GFP. Okay, and this is one construct, and in another construct, uh, another GFP construct generated using the deleted promoter region of 500 base pair starting from a transcription starting site. So this is basically TSS, and this is, for example, the 500 base pair site that is being removed. So now the site of promoter is reduced, okay, by 500 base pair. This is the transcription start site, and this is our GFP gene. So this is how the construct the question is talking about. Okay, so if suppose the promoter was of 2000 base pair here, in this case it has been reduced to 1500 base pair. Okay, and the region that is removed is from 1000 to 1500 base pair. So suppose this region has been deleted in case of in second example. Okay, 500 base pair from the middle part has been removed. Now the question is saying, so this vector is named as A. Okay, okay, there's no name to the other one. So the question is saying that there is the GFP expression in both the construct is same. So what can be inferred from this experiment? Okay, so this is the construct that we are talking about. Now let's understand the inference if the GFP expression under both promoter is basically same. So now, if 500 base pair upstream of TSS from any promoter is removed, and despite that the GFP expression remains same, it will mean that that particular region from that promoter is not essential for the transcription activity. Okay, and secondly, it will not contain any essential regulatory elements like some core promoter elements or some necessary enhancements, and that is why it is not affecting the transcription of the downstream gene, which is GFP. Okay, so the correct interpretation from this experiment will be, while doing the promoter analysis, that so how you can do it, that if the deletion of a particular fragment of promoter is reducing the expression, then in that case that fragment will be working as a repressor or as an activator, but that region will be important because it is changing, even reducing or even increasing in both the cases. If the removal of any fragment from the promoter is causing any change in the expression of the downstream gene, that means that particular region in the promoter is important. But if the deletion is not changing any expression, that means that part of the promoter is non-essential or it is an irrelevant sequence. So again, now about the question, if this is the promoter P and there is resulting GFP gene, and now from 100 to 1500 base pair sequence has been removed from this promoter, and this is the resulting GFP gene. But both this case have similar GFP expression. That means that region of the gene is irrelevant for gene expression. So it is not necessary, regardless whether that part of promoter is present or not present, the expression of GFP will still be same. Okay. So now analyzing all our options, that option A was region is responsible for coding gene. No, because if that was true, there should be some reduction in the expression of the GFP gene, and that is when we can say that the region was responsible for the coding gene. But that did not happen. So option A is also incorrect. Option B is the region is essential for expression of gene. No, because again, if this was the case, then the expression of GFP must have been reduced when that fragment was deleted. Option C is region is irrelevant for gene expression. It is correct. Yes, it is irrelevant because the removal is not causing any impact. And D is that region promotes RNA splicing. It has nothing to do with RNA splicing. The promoter regions have nothing to do with RNA splicing. So that is why in this case, option C is correct. Okay. I hope this was like you understood this question. It is just about the understanding of the concept of promoter and how the deletion assays actually work. Okay, let's move on to our next question. What is the most important feature of HpRNA constructs used in RNAi? Options are directly edit genes in the genome, contain only the coding sequence of the target protein, single stranded RNA formation without a secondary structure, and single stranded RNA formation that converts into a hairpin loop. Okay, so let's understand this question. So let's talk about first what is HpRNA. Okay, so HpRNA is basically short form for hairpin RNA, and hairpin RNA constructs are basically specifically designed for forming self-complementary RNA structures. And when these are transcribed, they will produce, after transcription, they will produce single-stranded RNA, but that single-stranded RNA will fold back. So basically, the RNA that is being produced is like this. Okay. But it immediately, once it is produced, it immediately gets converted to something like this, where the complementary part uh, becomes double-stranded and there is a small loop which is formed. Uh, basically a double stranded stem with a loop, which is this structure is called as hairpin structure. Okay, so this is what an HV RNA looks like. After production of this RNA, it is a single stranded RNA, but it will fold back to form a double stranded stem with a small loop. Now, this structure is why it is so important. So, this structure is crucial because in case of RNAi, the protein called dicer, which has to cleave this RNA into small fragments or siRNA called as small interfering RNAs. This can recognize only hairpin loop structure RNA. Okay, only when this kind of structure is present, only then dicer will attach to it, and then it will form these small interfering RNAs, which are responsible for RNA interference. Okay, so that is why the structure is important, and so this is given here that the structure is crucial because the double-stranded RNA is recognized by dicer, an enzyme which is used in the RNA pathway. And this dicer will basically process it into siRNAs or the small interfering RNAs, which will then guide the gene silencing. Okay, so this is the process of silencing. We have talked about this in previous classes also, that this is so if it has to be synthesized artificially by some promoter, then in that case the sequence that is being cloned is an antisense sequence, a sense sequence with a small linker sequence. So once this gets produced into mRNA, these two sequences will be complementary to each other, and the linker sequence will basically form the loop. Okay, so once this RNA gets produced, it will get converted into HpRNA, and this is recognized by dicer. Dicer will basically cleave the stem of this into small fragments called as small interfering fragments, small interfering RNAs, and this siRNA, along with RISC complex, will now be responsible for mRNA degradation or the RNAi process or RNAi interference silencing. And because now the mRNA, virus mRNA is degraded, there will be no resulting protein. Okay, this is the concept of RNAi. Okay, or RNA interference, silencing through RNA interference. Okay, so now according to our question, the options that were given was that option A was it directly edit genes in the genome. No, HpRNA has nothing to do with by in a direct edit causing direct editing in the genes of the genome. So it is incorrect. B was contains only the coding sequence of the target protein. No, it will also have a small um, loop, the sequence which has nothing to do with the target protein, to form a loop so that the dicer can identify. Option C is single stranded RNA formation without a secondary structure. No. The formation of hairpin loop is the secondary structure that has to be formed for it to be identified as HpRNA. And option D is single stranded RNA formation that converts into hairpin loop. Option D in this case is correct, that the RNA that is being produced will get converted into a hairpin loop like structure which has complementary sequences. Okay, so option D in this case is correct. Okay, so the next question is choose the correct combination from this table. And it is very easy. It is talking about the direction of the mRNA synthesis and the direction of the reading strand of the template. It is very easy question. You can try attempting it. Okay. So this is very easy question. We just need to know the direction of RNA synthesis and the direction of reading template. So this is the basic figure from which you can understand. Okay. So uh, transcription is basically conversion of from DNA strand to producing a mRNA strand. So our DNA is a double-stranded structure. So it will have two strands. One is from 3 dash to 5 dash, and the other one is from 5 dash to 3 dash. Okay. So now basically RNA polymerase is recruited to form the resulting RNA, and the resulting RNA will always be produced in 5 dash to 3 dash direction. Okay, it will always be from 5 dash to 3 dash direction. Now, because the direction is from 5 dash to 3 dash direction, and we know that the transcription or even DNA replication always happens on the opposite strand, that means the opposite strand must be 3 dash to 5 dash. Okay, so if the so in this case, the direction of transcription will be from 5 dash to 3 dash direction, and the direction of the reading template will be from 3 dash to 5 dash direction. Okay. So the correct explanation is RNA polymerase synthesis happens in 5 dash to 3 dash direction. And for this to happen, the resulting the used DNA template reading must be in 3 dash to 5 dash direction. And this basically anti-parallel mechanism will ensure proper base pairing and elongation of RNA strand. So based on this, our correct option is option B, because the direction of RNA strand synthesis is in 5 dash to 3 dash, and the reading strand is in 3 dash to 5 dash. Okay, so option B is correct. Now the next question is, if the coding strand sequence from 5 dash to 3 dash direction is ATG, CTG, ATAA, what would be the sequence of the transcript? Again, it is very easy. I'll give you 1 minute time. You can solve this. It's very easy. Okay. So let's solve this. The coding strand basically, or the sense strand, will have the same sequence as mRNA. Okay. So if the direction is in, so basically the mRNA direction, we have talked about it in previous question, that mRNA direction is in 5 dash to 3 dash. So the reading strand will be from 3 dash to 5 dash. But if you look carefully, the coding strand that is given here is in 5 dash to 3 dash. Okay. That means in case of DNA sequence, if this is 5 dash to 3 dash and this is 3 dash to 5 dash, then this must be read for the complementary mRNA to be produced. But if the fragment is 5 dash to 3 dash, then in this case, the sequence of the sense strand or the 5 dash to 3 dash strand will be very much same to the mRNA. Okay, the only difference will be that thymine in this case, because DNA contains thymine, but RNA contains uracil. So all the thymine in this case will be replaced by uracil. So this coding strand which is given will be get converted to 5 dash. So A will be as it is, T will get converted to U, G and C will be as it is. Okay, this is G, C. Again, T will get converted to U, G, A will be as it is. T again will get converted to U and A will be as it is. Okay. So this will be the resulting sequence in this case. So our option C in this case is correct. Okay. If it is sense strand or if the DNA sequence that is given is in 5 dash to 3 dash direction, then the resulting mRNA sequence will be exactly same except for the changes in place of thymine, it will get converted to uracil. But if the strand that is antisense strand or 3 dash to 5 dash strand is given, then in that case, the mRNA that will produce will be of complementary sequence. Okay, this you need to take care that if the coding sequence, if the DNA sequence that is given is from 3 dash to 5 dash direction, or it is called as antisense direction. Okay, then in that case, it will be complementary. Okay. So the next question is, which of the following is not a transcription factor? Options are CAAT, BZIP, BHLH, and Werky. Okay. So let's understand all the four terms in this. So first is cat. Okay. So cat is basically a small DNA sequence element, which is also called as cis regulatory element. Okay. And it is commonly called as cat box. And now where it is found, it is basically found in the promoter region of many eukaryotic genes, and it is present around 75, approximately 75 base pair upstream of the target start site. Okay. So whenever the gene is there, the target site of the gene. So first at around 30 base pair, a Tata box is present, and around at 75 base pair, the cat box is present. Okay. And the important function of cat box is that it will act as a binding site for various transcription factors. Okay. And it is not a protein, so therefore it is not a transcription factor. It is a DNA sequence that will allow the binding of a transcription factor, but cat itself is not a transcription factor. Second is BZIP or basic leucine zipper protein. Now, BZIP belongs to transcription factor family, and its structure is in such a way that it will bind, the basic structure will bind to a DNA, and the leucine zipper site of this transcription factor will allow the dimerization. Okay, and now the function of BZIP is that it will regulate certain gene expressions in processes like stress response, seed development, and metabolism. So example is HY5 transcription factor, which belongs to BZIP family, and it is involved in light signaling. Third is BHLH. BHLH stands for basic helix loop helix, and it is another huge transcription factor family, and its structure contains two alpha helices which are connected by a loop. And this is the basic region for DNA binding. The function is again in cell differentiation, development, and environmental responses. Example is MYC proteins in plants. There is another example which is MYB proteins in plants. This all belong to BHLH type. Okay. D is Werke. Werke is also a large transcription factor family which is very specific to plants. Okay, it is a plant specific transcription factor family, and its structure contains a conserved Werke domain which will bind specifically to the W box. Okay, now again, W box here is a small DNA sequence just like cat box. Except the fact that W box might or might not be present in the promoter region, but cat box is always present. Okay, so where if there is a W box sequence, whose sequence is also known, it is TTGACC. Okay, so this is the sequence of W box. So this sequence is known. So if this sequence is present in the promoter, Werke transcription factor can basically bind to W box and then regulate the gene expression. Now the function of Werke is that is it plays major role in both biotic and abiotic stress responses, in senescence and in defense signaling. Okay. Now, Werke is often studied in stress biology and crop resilience as well. So, according to the question, the cat box in this it will refer cat in this will refer to the cat box, and it is a cis regulatory element which is found in the promoter. It's not a protein, so it is not a transcription factor. On the other hand, all BZIP, HLH, and Werke are families of transcription factors. Which have functions in various binding of DNA, which will have the role of binding as well as of regulating the gene through its promoter. So the correct option to answer to this question is cat, that it is not a transcription factor. Rest all three are. Okay. Okay. So the ninth question is coding sequence of a gene was retrieved from NCBI and suitable primers were designed to amplify the target gene. Which of the following could be used for PCR amplification of the target gene? Options are genomic DNA, mRNA, plasmid, or cDNA. This also should be easy to solve. Please mute yourself. Okay. So let's try this question. Um, okay, again, it is a concept-based question. All right. So it is talking about coding sequence of a gene has been retrieved from NCBI. Okay. Now, when a question talks about coding sequence, that means that the sequence will not contain any DNA sequence of introns. Okay. The sequence will only contain exons, right? That is basically called as coding sequence of a region, and it will also not contain any promoters or any 3 dash or 5 dash sites or any other sequence other than the coding sequence. Okay. Now the primers have been designed to amplify the target gene, but what should be used as the template for this, for the PCR to amplify the target gene? Okay. uh, Now, all the four terms that are there in this case can be used to basically amplify. Okay, it can produce the PCR amplification of the target gene. But now let's talk about the tricky part to find the correct answer. Now, first option was genomic DNA. Now, why genomic DNA should not be used? Depending on whether your primer is how it is designed. Suppose this is your sequence of genomic DNA. Okay, that it will have one exon, intron, again exon, again intron, and again exon. Okay, this is the sequence of your genomic DNA. Okay, so yeah, okay. So the question is on your screen. You can read it from here. That the coding sequence of a gene was retrieved from NCBI, and suitable primers were designed to amplify the target gene. Which of the following could be used for PCR amplification of target gene? The options were genomic DNA, mRNA, plasmid, and cDNA. Okay. So as we were talking about genomic DNA. So now genomic DNA will have the sequences of introns as well as the sequences of exons, right? uh, in case of mRNA, that the mRNA or the cDNA, the coding gene that we are talking about, will have only the introns. Okay, this will be the coding. It will not exactly be called as cDNA, but it will be called as the coding sequence, or it is called as CDS actually. Okay, it is not cDNA; it is actually CDS. So this the CDS sequence will contain all the introns, all the exons joined together without any intron sequence. Now, if the primers are designed from this region, okay, that is, it will span from the first exon to the last exon, then in this case, both are genomic DNA and both the CDS. And CDS, the sequence is called as CDS, but our cDNA will actually be forming the CDS sequence. Okay. So in this case, both CDS and genomic DNA will give amplification. But the problem is that because the genomic DNA will also contain the intron part, there are chances that the amplicon size will be very, very higher as compared to the size that we are expecting. Because there are certain cases where suppose the genomic DNA size will span from 3000 base pair, but the gene size or the CDN or the CDS size is only suppose 1100 base pair. Okay, so in this case, if the genomic DNA is used as a template, first of all, the template size will be very high. Okay, the reason is that it will also amplify the introns. Okay, so that is why the amplicon size will be very high, and also there are chances that the PCR might get failed, and either it might get failed because also the exact amplification will not take place because there will be some added sequences in that. So in this case, from eukaryotes, the genomic DNA will contain introns, and the primers, if designed from CDS, either will cause PCR failure or the fragment size will be larger, as I was telling in this case. The PCR size will be much more than the expected size. So it is not appropriate to amplify CDS using genomic DNA. Now, option B was mRNA. Why mRNA is not correct? Because mRNA can never directly be used to form genomic DNA. Okay, because in case of PCRs, we normally use polymerase, DNA dependent DNA polymerase. Okay, so the Taq polymerase that is being used is a DNA dependent DNA polymerase. So it will require DNA as a template. Now, mRNA is not DNA, so it cannot directly be used for PCR. It has to be first converted into a DNA sequence, which is called as cDNA. Okay. cDNA will give the DNA as template, which can then be used for PCR amplification. So option B will be incorrect because it cannot directly be used for PCR. Option C was plasmid. Now, again, plasmid can be true if the CDS that we are talking about is already cloned in that plasmid. Okay. But if that gene is not cloned, then in that case, plasmid is useless for us. Only when the vector contains the sequence of the CDS that we require and the primers are designed from that, then in that case the amplification is possible. If not, then this option is incorrect. So because it is not very clearly mentioned in this, so it is not a reliable answer. Option D was cDNA. Now, cDNA will be the correct option. Why? Because cDNA is formed basically from the mRNA, and mRNA will always have only exons and no intervening sequences of introns. And once that mRNA is formed by using the process of reverse transcriptase, that also we have already talked about. Okay. So by using reverse transcriptase, it will get converted into cDNA. Okay. And cDNA will have sequences from uh, only from exons and no intervening sequences. And this sequence, the cDNA sequence will match exact sequence to the CDS which has been retrieved from the NCBI. So cDNA will be the correct template to ensure the amplification of our coding region that has been amplified from that has been retrieved from NCBI. Now, this is a small figure to explain how things work. DNA in this case is the genomic DNA, which gets converted into mRNA, which gets later converted to amino acid and then protein. So, this is the process, this is the genomic DNA. And when this genomic DNA gets when mRNA gets converted to cDNA, cDNA will have no introns and this is our plasmid. So if the target gene is the gene of our CDS, then in that case, plasmid can also be used as a template. But since it is not mentioned here, so plasmid is the wrong option. The correct option in this case is option D, which is cDNA. Okay. So this is the last question, and it is a little tricky question. So you need to understand this question well to be able to solve it. You can look at the figure. And this is also an example of promoter deletion assay that we talked about in first or second question in the beginning. And you need to know the concept of promoter deletion assay to be able to solve these kind of questions. Okay. So I will give you like 2 to 3 minutes. You can read this question because it is a time taking question. Yes, Yash, you have any doubt? Okay. So you can take 2 3 minutes. You can read this and understand this question, and you can try solving it. Otherwise, I'll explain the concept of this. Okay, somebody has answered option B. Anyone else wants to try? Yeah. Should we try solving this? All right. So, basically, in promoter deletion assay, let's first understand the concept of promoter deletion assay. So, in this case, the fragments of promoter, which is deriving a gene, is removed part by part to understand how it is regulating the downstream gene. Okay, so there are three possibilities that can happen. If a fragment, when it is removed, is changing the gene expression, okay, it can either increase or decrease, then it means that that particular fragment in the promoter is relevant and it is necessary in case of the downstream gene. Okay. The second possibility is that by removing that fragment, there is no change in the downstream gene expression. Okay. So if there is no change, then in that case, that particular fragment is not necessary and it is irrelevant in case of gene expression. Now, the first possibility that I talked about, that there is a change in expression, there can be two things. That if there is a change in expression, it can either increase or it can cause decrease. Okay. Now, if the expression due to removal is increase in the gene expression, that means that fragment is acting as a repressor. Okay. But on the other hand, if the change in the gene expression is decreased due to removal of that particular fragment, then that fragment is acting as an activator. Okay. So these can be three possibilities, that whether that the fragment of the promoter is acting as a repressor, whether it is acting as an activator, or whether it has no function in regulating the downstream gene. Okay, I hope this concept of promoter deletion assay is clear. Now, coming back to our question, let's just take the figure and understand this. That in this case, the part A is a full length fragment, okay, of a 1250 base pair is a full length fragment. In part B, from 1250 to 1000, the fragment has been removed. In C, from 1000 to 750 has been removed. In C, from 750 to 500. And in D, from 500 to 250 fragment has been removed. Okay. If we are saying that this fragment has been removed, that means the entire promoter will consist of this plus this sequence. Okay, these two sequence get joined and the middle part gets removed. Now, because this is a promoter, there has to be a downstream gene. Okay, and this downstream gene expression is given in this graph. Now, if we talk about A, A fragment, okay, so A fragment is showing 75% of gene expression. That means full length promoter, A is the full length promoter, is showing 75% of gene expression. B is where the first part of the fragment is getting removed. Okay, first 1250 to 1000. Okay, so that means 250 base pair is getting removed, but the gene expression is same. That is 75%. That means that the B fragment has no relevance in case of the downstream regulation or the downstream expression of that gene. Okay. Now C fragment. C fragment is where from 1000 to 750 base pair, 250 base pair has been removed. Okay. Now if you look at C fragment expression, the expression is 100%. That means the expression from the full promoter to that 250 base pair being removed has increased. Okay, this is showing an increase in expression. So that means that the fragment C is acting as a, in this last case that we talked about, that when the expression is increased, it is acting as a repressor. Okay, that means the presence of that particular fragment is causing a reduction in the gene expression. That is why when that fragment is removed, the gene expression has increased. So in this case, fragment C, that is the fragment from 1000 to 750 base pair, is acting as a repressor. Now let's talk about fragment D. Okay, so fragment D, again the expression level is 75% only. So that means again it has no relevance. It has nothing to do with the downstream gene expression. And fragment E, which is from 500 to 250 base pair, the expression is zero. Okay, that means that fragment E is very essential for the transcription of downstream gene, and also it will act as an activator. Okay, that means that the presence of that fragment will basically regulate the expression of the downstream gene. Understood? This is how you solve these kind of questions when the question is about promoter deletion assay. Now let's talk about the options that was given. So option A was that region 1250 to 1000, which was B fragment. Okay, houses positive regular. Here we found out that our fragment C was a repressor, and fragment D was a C was repressor, and fragment D was an activator. Okay. Now option A says that B is a positive regulator, and from 750 to 500 it was D. So B and D, B is positive and D is negative. So this is incorrect. Option B says that D is positive and E is negative. Again, it is incorrect. Okay. Option C says that 500 to 250, that means E. Okay. E is a positive regulator. That means it's an activator. And C is a negative regulator. So yes, this is exactly what we found out, that C is a negative regulator. That is, it is a repressor. And D is a positive regulator. That is, it is an activator. So in this case, option C will be correct. Option D was 750 to 500. That is, E is a positive regulator, but 250 to 1 is a negative regulator. So we don't know about 250 to 1 because that particular part it has not been talked about in the question. So we cannot basically rely on that answer. So option C in this case will be correct. Okay. I hope this was clear. This question, I know it's a little difficult to understand, but if you have the concept of promoter deletion assays clear, it is very easy to solve this kind of question. If you follow this methodology in the exam, it will become easy for you to solve it. Okay. Okay, if anyone has any doubt in this, you can ask. Otherwise, we can end this session.